A very good evening to all of you present over here. So today, myself, I am Dr. Violet. So I'll be discussing the questions from Unit 7. So basically, we'll be discussing the questions from the topic. So these are the CSIR NET questions. That is from endocrinology and reproduction, as well as from nervous system. Okay. So the first question that we can see over here is about the endocrine system. Okay. So before I start with the questions, please confirm if I am audible, if the slides are visible. All good? Yes, Vishali. It will be uploaded. It will be there. The session recording will be there on the YouTube. You can get access to it anytime you want. Okay, thank you, Anjal, for confirming. Okay, thank you, students. So the first question, as I said, it is from endocrinology. Now, this is a part C question. Okay, so we'll be discussing both of it. So you get an idea of how the part C as well as part B questions are asked. So this is a statement-based question. So it's about testosterone. It says is a steroid hormone about which a set of statements are given below. Yes, Sunshine, good evening. So the statements that are mentioned over here, statement A is it is secreted by Leydig cells of testis. B, it is secreted by Sertoli cells of testis. C, it inhibits secretion of LH, that is the luteinizing hormone, from anterior pituitary. D, it does not inhibit secretion of FSH, that is follicle stimulating hormone, from anterior pituitary. So which one of the following combination represents correct statements from testosterone? So you have to search for the correct statements. Keep this in mind and just try and let me know what should be the right answer. I'll give you some time. So I'm sure you all might have started preparing for the upcoming exams. So this question you all can try and tell me. It's not very difficult. It is simple, just that it looks big because certain statements are written. Okay, Reshma, I think you need to try again. You are correct with 1 point, that is correct. Okay, the one statement is correct. The other one is not correct. Yes, Karen, Sunshine, Anchal. So your answers are correct. So the right answer, okay, many students are still, yes, Reshma, now it's correct. Very good. So the right answer over here, okay, students are still answering. Yes, Shayantani, correct. Good. So the right answer over here will be option 1. Okay. So what is the option 1 state? Option 1. Option 1 is statement A and C. Right. Now testosterone is secreted by Leydig cells. Remember. Okay. Where are the Leydig cells present? Inside the testis. Okay. When you see the cross section. So they lie between the seminiferous tubules. So inside the testis, you have the tube-like structures, right? And in between them, you will have the Leydig cells that will be present. So those are the Leydig cells. And testosterone is secreted by cells that are lying between the seminiferous tubules that are connected to each other, right? And this testosterone, it is No, it can inhibit the release of LH. So LH release is how LH is being released. LH is luteinizing hormone, right? It is released from anterior pituitary. Now, what will stimulate the release of LH? It is the gonadotrophin releasing hormone. From where the gonadotrophin releasing hormone is being released? It is released from hypothalamus, right? So hypothalamus will release the gonadotrophin releasing hormone that will stimulate the anterior pituitary to release LH, right? And LH will in turn, it will stimulate the Leydig cells to release testosterone. Okay, so LH release is stimulated by gonadotrophin releasing hormone, and it can be inhibited by estrogen in females as well as testosterone in males. So testosterone can inhibit the release of the LH. Okay, and if you talk about females, so their estrogen can inhibit the release of LH. Yes, Anu, did you get it? Okay, so the right answer is option 1, not 4. Not sure why everybody is okay. Right? So, what is the function of Sertoli cells? Do you all know? No, it's very important to know the function of each and every cell that is mentioned. So, what is the function of Sertoli cells? What will Sertoli cells do? Nutrition, very good. So, basically, it will help in spermatogenesis. Okay, so nutrition for what? For the development of sperms, that is spermatogenesis. Correct, Karen, Reshma, correct. Okay, nourishing the development of sperm, very good. So it helps in spermatogenesis. So the development of the sperms that is taking place. So for that we need the Sertoli cells. Okay, Leydig cells we understood, it is for the release of the testosterone. Okay, so testosterone will release Leydig cells. And then we saw the other statements as well. It inhibits the secretion of luteinizing hormone from anterior pituitary. This is correct, and option A is correct. Okay, so let's move on and see the next question. Okay, again this question is from endocrinology part. Yes, sunshine, correct, for spermatogenesis, absolutely. So given below are some statements about pituitary hormones. Oxytocin and vasopressin are synthesized in posterior pituitary. Prolactin is synthesized from anterior pituitary. Alpha and beta MSH are secreted from intermediate lobe of pituitary in adult humans. D. Growth hormone secretion from anterior pituitary is stimulated by hyperglycemia. E. Prolactin secretion is markedly increased by sleep. So choose the incorrect combination of statements from below. So remember, you have to select the incorrect statements over here. Okay, so try and tell me what should be the answer for this particular question. So basically all these are pituitary hormones. So you need to know in details about the pituitary hormones. Okay, like FSH, LH, all the details you need to know. Oxytocin. Okay, then we have vasopressin. We have so many different other hormones. MSH. So what should be the right answer? Okay. Okay. So many of you all are answering, but the answer is not the correct answer. Try again, students. You have to find the incorrect ones. Okay, Karen is correct. Absolutely, very good. You come with good preparation. Vanishri, Ashwarya, somebody named Beautiful Colors. Priyanka, you need to try again. Anu, Prachi, Rohit, try again. Incorrect, students. Many of you all are saying option 3. Those are incorrect. Do you all think statement A is correct? Sunshine, correct. Very good. Reshma, you answered not yet, right? That was for previous. Anu, correct. So if I say A is not correct, then I think many of you all should get the right answer now. If A is not correct, then we have two options where A is present. And if you know about B, so simply, these type of questions, if you know one or two statements, it's easy to find the right answer. Yes, very good, Karen. Oxytocin and ADH, they are synthesized where? Is it posterior pituitary? Are they synthesized in posterior pituitary? Why I said it is incorrect? Where are they synthesized? Kavyarisi, good try, but you need to try again. Option 2 is not the right answer. Okay, the right answer, students, it is option 1. Okay, so now let us see each and every statements. Oxytocin and vasopressin, they are synthesized in posterior pituitary. I said no, they are not synthesized in posterior pituitary. Then where they are synthesized? Where you will find them? Yes, right, Ashwarya. So in the hypothalamus, correct. So they are synthesized in the hypothalamus. So remember that, students. Okay, and they'll go and get stored in posterior pituitary. Okay, so these details is very important to know. Then prolactin is synthesized by anterior pituitary. Definitely B is correct. So if you know A is incorrect, B is correct, then definitely this is not the option. This is not the option because B is present, and A is the option because A is incorrect. So like that, you can find out what is the right answer. You need not know other statements as well. Okay. So if you know A is incorrect and B is correct, then you have one option over here like that. So it is A, C, D, right? Easy to solve that way. So many times, statement-based questions, though they look tricky, but if you know one or two statements, it becomes easy to solve them. Okay. So there is no dilemma. You will not be in a dilemma because you are confirmed with the two statements. Okay. So in this option, A is incorrect, B is not present means this is the most probable one, right? So A, C, D. So B is a correct statement. As I said, prolactin is synthesized from anterior pituitary. Then C is about alpha and beta MSH are secreted from intermediate lobe of pituitary in adult humans. Now why C is incorrect? Definitely, I think I have told you all that alpha and beta is secreted from pituitary, but it's not the intermediate lobe. Okay, so when I remember we have studied this part and then we were discussing where it is being synthesized. It is definitely synthesized from the anterior pituitary. Okay, but intermediate lobe is actually during development it will be present. Okay, in the not you'll not see this intermediate lobe in the adult humans. So this is why this statement is incorrect. Okay, so the adult human pituitary does not have a distinct intermediate lobe. and therefore it is not a source of alpha MSH, in particular about alpha MSH. So alpha MSH is not thought to be produced in the anterior lobe, but it is not very sure, where from where exactly it is being produced. But yeah, as we have also discussed, this MSH is not very sure about where it is being synthesized. Okay, but definitely it is not intermediate lobe because it is not present in the adult humans. Okay, so that is why C is incorrect. Now, D, growth hormone secretion from anterior pituitary is stimulated by hyperglycemia. Will it be influenced by the involvement of glucose? No, right? Hyperglycemia means more glucose in the body. Will it stimulate growth hormone secretion? No. So this is again an incorrect statement. Okay. So the right answer over here is option 1, A, C, and D. Okay. And about this prolactin secretion. Okay, so can you all tell if E is correct or not? What stimulates the prolactin secretion? It is correct. So basically, the prolactin secretion is during the pregnancy. Okay, so it is, to certain extents, the sleep cycle will influence it. Okay, so during the sleep cycles, there are certain periods. So during which, if the sleep is being deprived and all that, so the prolactin secretion will get inhibited. Okay, so that is why C, the E statement is correct. Yes, right, Vanishri, correct. Susan, melanin? Yes, melanin is a pigment. Okay, so MSH will further, this hormone will go and stimulate the melanocytes. What are melanocytes? Melanocytes are those cells that are present, okay, in our body that will release the pigment that is melanin. Okay, so the pigmentation is occurring because of melanin. So what is melanin? It is a pigment. Clear? So the skin darkening that occurs. So it is because of, if more the melanin, more darkened the skin becomes. If less melanin, less, little brighter. Clear? Okay, so let's move ahead. So already I have discussed about oxytocin and vasopressin. So they are neuropeptides synthesized in the hypothalamus and secreted from posterior pituitary gland. Okay, and other points we have already discussed. Okay, next question is from nervous system. The nervous system is also very important from CSIR point of view. Okay, nervous system as well as endocrinology, they are very important. You will definitely see one part C question that will come from both, one from nervous system. Okay, so the pattern is such, like at least 4 part C you will see for unit 7 at least. Okay, more can also be present. Usually one from nervous system. Okay, then one from endocrinology and reproduction, one from cardiovascular system, and sometimes they might ask you from temperature regulation, or they can even ask you from respiration. Okay, usually before they used to ask from sense organs. Nowadays I'm not seeing much questions from sense organs. So these are some of the important topics that you need to go through: nervous system, endocrinology, cardiovascular system. Okay, then temperature regulation, nowadays they have started asking questions related to that. So you should be very thorough with these topics, as well as respiration. That is important part. So let's see this particular question. Following are the statements made about major functions of some of the neuroglia in normal condition. A. Oligodendrocytes help maintain potassium level and contribute to the blood-brain barrier. B. Microglia capable of movement and phagocytosis of pathogens and damaged tissue. C. Astrocytes produce the myelin sheath to electrically insulate neurons of the central nervous system. D. Ependymal cells, which are ciliated, are involved in circulation of cerebrospinal fluid. Which of the following options represents the combination of all incorrect statements? So can you all try and tell me? No. Try again, students. You have to find incorrect statements. Okay. Anupam, Sunshine, Karen, try again. Reshma, Priyanka, Ashwarya. Correct. That is correct. Very good. Susan, try again. Yes. Karen, correct. Yes, many students have answered. Good to see so many of you are answering. Uh, Saujanya, correct. Very good. Are you all able to solve? You all know what is neuroglia cells? Right? So neuroglia cells, what they are? They are not neurons, right? They are the cells that are present along with the neurons, and they are more in number, and they have some particular characteristic. They are different types. So if you talk about central nervous system, so it has 4 different types of neuroglial cells. If you talk about peripheral nervous system, it has 2 different types of neuroglial cells. Yes, Ashwarya, correct. Option, what you are saying? No, all, no. Yes, yes, right. Correct. Absolutely, Ashwarya. I agree with you. Okay. So you are absolutely right. So oligodendrocyte is a neuroglial cell for the central nervous system. Okay. It maintains the potassium level. No. So oligodendrocyte is a neuroglia of the central nervous system. But what is its major function? Okay. It does not contribute to this blood-brain barrier. What it does basically is it helps to form the myelin sheath of the neurons of the central nervous system. Remember that. That is why A is incorrect. Okay, students? Yes, Amrita, correct. Very good. Yes, Reshma. Yeah. So basically, they form the myelin sheath, the oligodendrocytes. Okay. Microglia, they are capable of movement and phagocytosis of pathogens and damaged tissue. So B is correct. Okay, but they have asked for incorrect. That is what we have to find. Okay, sometimes it happens that we select the correct ones because we are so you know, we always practice for correct statements, so that is why we make a mistake. But you have to see the questions properly. So it is written incorrect, so A is incorrect. So the options can be 1, 2 or 3. Now next, B is not correct, so definitely this is not the answer. Let's see C. Astrocytes produce the myelin sheath to electrically insulate neurons of the central nervous system. No, myelin sheath is not produced by the astrocytes. Okay, what is astrocytes function? Can you all tell me? C is incorrect. Okay, so here we got it. A and C are incorrect statements. But what is the actual function of astrocytes? What it does? Any major function you all remember? Astrocytes is very important. Okay, neuroglial cells that performs a large number of functions. But any one important function that you remember? Yes, Saujanya, very good. So one of the important functions that here you can see it helps to maintain the blood-brain barrier. Okay, so that is an important function. Neuronal activity, protect neurons, correct. Learning, memory, brain development, if you'll remember, homeostasis, right, Sunshine, correct. So the right answer over here is option 2, A and C. So C is an incorrect statement. Why? Because they have written myelin sheath. Myelin sheath is not produced by astrocytes. Okay. So for peripheral nervous system, which cell will form the myelin sheath? Yes, Priyanka, correct. Isolate neurons from harmful. Yes, very good. So for peripheral nervous system, which cell forms the myelin sheath? For peripheral nervous system. Schwann cells, correct, Karen. Right. Beautiful colors, correct. So that is what we have to remember each and every point related to the different types of cells, correct? Reshma, Priyanka, right. Okay, the D statement, it says ependymal cells which are ciliated involved in circulation of cerebrospinal fluid. So that is correct, right? Ependymal cells, they'll have cilia and then they line the ventricles. Okay, and from it also helps in the circulation of cerebrospinal fluid and even the exchange of materials that can take place. So that is through this ependymal cells. Okay. So same thing is mentioned over here. Let me quickly go through it. Oligodendrocytes are myelinating cells of central nervous system. Astrocytes are a type of glial cells. So they are more in number. So they make up the majority of cells in central nervous system. They perform metabolic, structural, homeostatic, and neuroprotective tasks like clearing excess neurotransmitters, stabilizing and regulating the blood-brain barrier, and helping in the synapse formation. Microglial cells, they are a specialized population of macrophages found in the central nervous system. They remove any kind of damaged neurons and infections, and they're important for maintaining the health of central nervous system. Okay, so these are the points that you need to keep it in mind. Now the next question is again from nervous system. So it says an experimentalist stimulates a nerve fiber in the middle of axon and records the following observations. Which one of the observation is correct? Nerve impulse is traveling in a direction towards cell body. Nerve impulse is traveling in a direction towards telodendrons. Nerve impulses are traveling in both the directions opposite to each other. Nerve impulse is not moving in either direction. So what do you all think? What should be the right answer over here? Vanishri, try again. What is a telodendron? Now, if you have seen the structure of a neuron, so this is the dendrites, right? This is the cell body. Then you have the axon. Then axon will end into axon terminals, right? Something like this. So these axon terminals, so these are your telodendrons. Okay. Yes, Anupam, Tonio, correct. Amrita, correct. Saujanya, correct. Reshma, Dimat, Karen, Sunshine, Susan, you need to try again. Just think properly. Balambal, that is not correct. You need to try again. I think during no, when we were discussing the movement of action potentials, I had mentioned this part, how the nerve impulse will travel, in which direction. Yes, Demat, correct. Priyanka, correct. Very good. Okay, so let's see the answer for this particular question. Answer is option 3. Okay, so what is option 3 over here? It says Nerve impulses are traveling in both the directions opposite to each other. Now, if the conduction, if it gets stimulated over here, then definitely the conduction will move like this. Okay. If it gets stimulated in the middle, then what will happen? The conduction can move in this direction also, conduction can move in this direction also. They are the end terminals. Okay, Ashwarya, what are telodendrons? They are the end terminals of the axon. Okay, so those are termed as telodendrons. Okay, these end terminals, these are the telodendrons. Axon terminals also you can say. Right? So if it is stimulated in the center, the conduction can move in both the directions. Okay, if it is over here in the cell body, then definitely it will move in this direction. Okay, so now here when one conduction is moving, definitely another conduction cannot move. Okay, so nerve impulse is not moving in either direction, that is incorrect. Nerve impulse is traveling in a direction towards cell body, so that is also not correct because the conduction will not move only in one direction. Okay, that is why one is incorrect. Two, nerve impulse is traveling in a direction towards telodendrons, so this is also not correct. So it will move in both the different directions because what they have mentioned in the question is it is stimulated in the middle of the axon. So if you stimulate in the middle, it can move in both directions. But if you stimulate over here, it can only move in one direction. Why it can move in one direction? Okay, Ashwarya. Sunshine Vanishri got it. Okay. So why it can move in one direction? Why not? It can move into two different directions at the same time. Can you all think of anything? If maybe if it has got stimulated over here, the conduction is moving in this direction, and again if something has stimulated over here, can it move like this? No, right? Two conductions cannot move at the same time. Why? Any idea? So this is the figure about the neurons. Okay, and this is how the action potential passes. So in response to a signal, the soma end of axon will become depolarized. So this depolarization will spread down the axon. Okay, because here it is in the soma, the cell body. So that is why it will move like this. So meanwhile, the first part of the membrane will repolarize. So once it stimulates the next region, so this part will repolarize. The other part is getting depolarized because sodium channels are inactivated and additional potassium channels have opened over here. The membrane cannot depolarize again. In the repolarizing phase, the potassium channels have opened and sodium has got closed. Okay, so once this has happened, the membrane cannot depolarize again because it will be in the refractory period. Not graded potential, it is about action potentials, okay, but it will be in the refractory period. Okay, it is not about distance because once it is, if you'll remember, we had discussed about the sodium channels. Okay, so they have two gates. Okay, so when they have two gates, so the sodium will initially be in an inactivated state. So they have to return back to the activated state. So when they have to return back to the activated state, so that time only another action potential will pass. Okay, so now if it has got depolarized and it is in the repolarizing phase, so for some time it is in the inactivated state, the sodium. So that time another action potential cannot be passed. Okay, only it reaches back to the activated state or the resting state, then only it is possible, right? So that is why another action potential cannot pass at the same time. So the action potential will continue to travel down the axon only. Okay, because again this will be in the repolarizing phase. Okay, then depolarization. Clear? So that is why at the same time two different action potentials cannot pass. Clear for everyone till here? So before I go on to the next question, I would like to talk about the Raftar batch. So this is the batch for December 2023 exam. It is a very what we can say is a very important batch that everybody should join. So it is a speed revision batch for CSIR NET December 2023 exam and it will be starting from 11 September. So here there will be, topic coverage. So any of your friends, your juniors, seniors who are interested in this uh, topic coverage, guarantee will also be given for Avatar batch. So all the important topics that will be asked in December 2023, so that will be covered up. You'll also get access to the study portal as well as the PowerPoint presentations. Okay, all the classes in which the PPTs are being used. So those PPTs will be shared. Daily practice sheets will be presented. Okay, it will be shared with the students, as well as they'll have 24/7 faculty support. They can ask questions uh, at the chat, and they'll be provided with 100 plus animations. So they'll have access to this animatica zone, as well as they'll have access to the AIMNet test series and the different revision tools that are very popular in Biotechnica. You might have seen many of it and they are very useful. So anybody is interested, they can join this Raftar batch. So you can call this toll free number 1800 or 08050997000 for joining the Raftar batch and more to get more details about it. Now moving on to the next question. So the next question is again related to the nervous system. So it says in the resting state of a neuron, the axonal membrane is, you have to tell the right statement, okay? What is happening to the axonal membrane? It is comparatively more permeable to potassium ions and nearly impermeable to sodium ions. It is comparatively more permeable to sodium ions, more permeable to sodium ions and nearly impermeable to potassium ions, equally permeable to both sodium and potassium ions, impermeable to both sodium and potassium ions. So can you all think and tell me? Okay, just now we discussed about the neurons. So which statement is the correct answer over here? So think about it. It is the resting state of a neuron. Very good. Sunshine, Vanishri, Ashwarya, superb. Everybody has given me the right answer. Demat, Ruben. Okay. Yes, Priyanka, Anupam. Correct. So let's see this question. So whenever the neuron is in the resting state, always remember it is more permeable to potassium ions. Okay, and nearly impermeable to sodium ions. So the right answer, yes, Amrita, you are also right. So the right answer is option 1, students. Okay. So here, what is the option 1 about? It is saying that when the neuron is in the resting state, okay, so you can see it is more permeable to potassium. And what is happening to the potassium? Potassium will move out of the neurons. Okay, and the number of sodium that is entering is less. So it is less permeable to sodium, but it is more permeable to potassium. Okay, so or you can say it is nearly impermeable to sodium. Clear? Okay. So because potassium is moving out, less sodium is entering inside, and then we have the sodium potassium pumps. Okay, and how they work, we know with the hydrolysis of ATP, 3 sodium moves out, 2 potassium moves in. So all because of all these kind of changes where positive is going outside, okay, so inside is more negative. So inside of the membrane is negative. So it gives a resting membrane potential of around -60 or -70 also, depending what type of cell it is exactly. Okay. Tell me, does all of the cells have the same resting membrane potential? What do you all think? Do they have the same resting membrane potential? No, it differs, right, Ashwarya, Priyanka, Sanshankar? Because each cell is different. Their number of the channels that are present, okay, the ion channels, they might vary. Okay, so because of all that, and then what type, how many ions are moving inside, okay, whether it is positive, negative. So based on all these factors, the resting membrane potential will also vary. Okay. So when a neuron is not conducting any impulse, remember it is in the resting state. So the axonal membrane is comparatively more permeable to potassium ions and nearly impermeable to sodium ions. Now the question number 6. Yes, Vanishri, right. Okay, it's a part C question. Okay, that was asked in previous year question papers in CSIR. So it is all about nervous system. So the membrane potential in a giant squid axon recorded intracellularly at the resting condition of minus 70 millivolt was reversed at the peak of action potential plus 35 millivolt after stimulation of the nerve fiber with a threshold electrical stimulus. This overshoot of the membrane potential has been explained in the following proposed statements. A rapid increase in sodium conductance during early phase of action potential uses membrane potential to move towards the equilibrium potential of sodium, that is plus 45 millivolt. B. The sodium conductance phase quickly decreases toward resting level after peak in the early, and sodium ions are not able to attain its equilibrium potential within this short time period. C. The conductance of potassium at the early phase of action potential is increased, and that leads to the reversal of membrane potential. D. The increase of potassium conductance due to stimulation of nerve occurs before the changes of sodium conductance is initiated and thus causes overshoot at the peak of action potential. So you have to find out which one of the following is correct. So you can read again and just think properly and try to answer. Nobody is able to solve. Okay, Vanishri has tried. Try again. That is correct, but one more is there. Okay, Vanishri. What option you said? So that statement is correct, but one more statement is also correct over here. Yes, Ashwarya, very good. Right. Anupam, Vanishri, correct, no? So what it says, this particular question, the resting condition is minus 70 millivolt, okay, and then depolarization will occur, it reaches plus 35 and then it will reverse back, okay, means like this, okay, depolarization and repolarization. So depolarization at minus 35, sorry, plus 35 and it is at minus 70 millivolt. So the first statement, okay, still students are trying. Yes, Ruben, Sunshine, correct. Priyanka, Karen, very good. Correct answers. So the right answer over here is option B, students. Okay. So let's see the first statement. What is B? B says A and B is correct. So the rapid increase in sodium conductance during early phase of action potential uses membrane potential to move towards the equilibrium potential of sodium. So yes, that is what is happening. So once from minus 70 it reaches the threshold of around minus 55, depolarization occurs and sodium will enter inside. So there will be change in the membrane potential. So the membrane potential will it will try to reach the equilibrium potential of sodium, that is around plus 45, but it will not be able to do that. Okay, before that only the sodium channels will close. So that is why A is a correct statement over here. Okay, so it does try to reach. Okay, it uses membrane potential to move towards the equilibrium potential of sodium, but it will not happen like that. But definitely it will try to reach towards the equilibrium potential of sodium. Okay, so A is correct. B, the sodium conductance phase quickly decreases towards resting level after peak in the early phase, and sodium ions are not able to attain its equilibrium potential within this short time. So that is what I told you again. The same thing, they have just continued the statements that sodium ions are not able to attain its equilibrium potential, right? So that is why the option B is again correct over here. Okay, now see what it says. The conductance of potassium at the early phase of action potential is increased, and that leads to the reversal of action potential. No, in the early phase there is no conductance of potassium as such. Okay, so early phase is depolarization, right? So there is no conductance of potassium. So that is why there is no conductance of that is being increased over here. That increases this action potential. So C is incorrect. D, what it says, the increase of potassium conductance due to stimulation of nerve occurs before the changes of sodium conductance is initiated. Is it occurring before? No, right? First it is sodium only. After that only potassium channels will open. So again D is incorrect. So the right answer will be A and B. Option B will be correct over here. Okay, so this figure is about the different phases. You can see the neurons will have a resting membrane potential of minus 70 millivolt. So whenever a stimulus comes, it will get there will be a change in membrane potential. It will depolarize. It will reach a threshold of around minus 55. That time only action potential will be generated. So initially it is a depolarizing phase. Okay, it will try to reach towards the equilibrium potential of around plus 45 millivolt, but it will not go towards that. At around plus 30, it will undergo the repolarizing phase. So sodium channels will close over here, and then potassium channels will open. Okay, so that is why it will not continue reaching towards the equilibrium potential. It will, it will form the repolarizing phase. Okay, and now in repolarizing phase, potassium will move outside. So that is why from positive, the value will go towards negative. Okay, but does not stop over there because more and more potassium channels will be open. So that will make it more negative. Okay, after hyperpolarizing phase will be present. Okay, so that was the sixth question. So this type of questions are very important from action potentials. Okay, you need to know this graph thoroughly. So here you can see it's already mentioned about the different channels. Sodium channels are inactivating over here. Okay, then sodium channels will return back to the resting state. So there is the refractory periods that are present. Okay, so this green to pink in this region, this is the refractory period. So once they are now once action potential is generated, so it is a refractory period, absolute refractory period, where another action potential cannot be generated. Okay, with a normal average stimulus strength. But after that, you have another refractory period that is known as relative refractory period. So that will fall in this, okay, this orange color. So in this time period, another action potential can be generated, okay, because the sodium they have returned back to the resting state. So you can see sodium channels are in the resting state. So that time there's a possibility that uh, the another action potential can be generated, but the stimulus strength has to be very high. Okay, the next question is question number 7. The afferent nerve fibers of a stretch reflex were electrically stimulated and the contraction of the muscle innervated by efferent fibers was recorded. The synaptic delay was calculated from the time points of the nerve stimulation and response of the muscle. Which one of the following time durations will be probable value for observed synaptic delay? So can you all tell me? Okay, first tell me what is synaptic delay actually? What do you understand by synaptic delay? What is synaptic delay? Any idea? What is this synaptic delay? Nobody knows? Okay. So now let us first talk about synapse. What is synapse? So if you imagine if this is a presynaptic neuron, okay, so this is the end terminal of the presynaptic neuron, and this is the dendrite of the postsynaptic neuron. Okay, so what is this synaptic delay? So it is the time taken, okay, for the neurotransmitters from this neuron. So first these neurotransmitters will be released by the vesicles. Now those neurotransmitters will take some time, right, to cross this gap that is known as synaptic cleft. Okay, so they will take some time to cross this, and then they have to bind to the receptors that are present in the postsynaptic neuron, right? So that time period that it takes for the neurotransmitter. Yes. Okay, Vanishri. Okay. Difference between two inward currents. Not difference, it is the time difference. Oh, yes, Vanishri, correct now. Yes, sunshine, correct. So that time it takes, okay, so that is synaptic delay. So why we are saying there's a delay? Because it takes more time. So there are two types of synapses. One is electrical synapse, okay, and another one is chemical synapse. So this what they have given the question over here is about chemical synapse because it is between a muscle and innervated by fibers. So that time it is a chemical synapse. So for chemical synapse, the neurotransmitter will be released and it will pass through the synaptic cleft and go and bind to the receptor on the postsynaptic neuron. So there will be a delay. Okay. So that delay is known as synaptic delay. So now the question is how much delay occurs, when you compare electrical and chemical synapse. So electrical is much faster. Okay, so chemical has shows how much delay. So what is the time interval? So the time interval over here, sunshine, it's not 3, okay, it is 0.5 milliseconds. Clear? So it is in very short duration. Okay, seconds will be too much higher. So it is in milliseconds, 0.5 milliseconds. Yes, correct. Vanishri, sunshine, correct. Ruben, correct. Okay. So here you can see I have given you the figure of the electrical synapse and the chemical synapse. So this is a chemical synapse. So this is the end terminal of a presynaptic neuron and this is the postsynaptic neuron. So you can see in the vesicles there are neurotransmitters. So once the appropriate signaling occurs, so calcium will enter inside, that is the signal. So there will be exocytosis of these vesicles. So they'll release the neurotransmitter, it will pass through the synaptic cleft and go and bind to the receptors. And then the current will pass in the next neuron. So that time period, that delay. Now, if you compare electrical, so quickly the current will pass from one cell to other cell because they are connected to each other through gap junctions. Okay, so here it is much quick, or you can say it is much faster, but here it is slower. So there's a delay. Okay, because all this process will take time, releasing, binding. Okay, Sunshine, good. You know it, that is very good. Okay, so that there will be a delay. So the delay is around 0.5 milliseconds. Right? So now let's move ahead and see the next question. So this will be the last question for the day. Which one of the following neurotransmitters is secreted by the preganglionic neurons of sympathetic nervous system? So can you all try and tell me? I think this was a part B questions. So they ask questions from neurotransmitters as well. Okay, so you need to study that as well. Sympathetic nervous system. Okay, Anu is very quick. Very good. Right answer. Anupam, very good. I thought this will be difficult. You are too quick. Very smart students. Yes, Amrita, right. Everybody has given me the right answer. Ruben, correct. So the right answer over here, it will be option 2, students. That is the acetylcholine. Okay. So I'll give you understanding of this particular type of questions that they have asked over here. So they have asked which neurotransmitter is secreted by the preganglionic neurons of sympathetic nervous system. So next time they can ask you about parasympathetic, okay, or they can ask you about postganglionic. Okay, so anything they can vary. So it's important that we understand, and you need to know this figure as well. Okay. So autonomic nervous system is again divided into two parts, right? Sympathetic and parasympathetic. Sympathetic is associated with fight or flight responses. Okay, so this fight or flight response will get activated when the sympathetic division is activated. While the parasympathetic, this controls the rest and digest activity, means mostly when the body is at rest. Now this is a preganglionic neuron. Okay, so the first one is preganglionic. Next neuron to which the impulse moves. So the next neuron is known as postganglionic neuron. Okay. Now it depends whether it is a preganglionic of parasympathetic or a sympathetic. So based on that, the neurotransmitters are released. Okay. So this preganglionic neuron soma, usually it is in the spinal cord. Okay. And the neurotransmitter that is released is acetylcholine usually or sympathetic. While for parasympathetic, the soma is usually in the brain. Okay, not in the spine, or sometimes the last portion of spine. Okay, sacral part. So this type of preganglionic neuron that is present in parasympathetic nervous system. Okay. So here preganglionic synapse for both parasympathetic and sympathetic, they'll release acetylcholine. Remember. Okay. Then next will the postganglionic neuron. So soma is usually in a ganglion near the target organ. So here you can see this is the cell body and it is very near to the target organ. So it is not very long, the axon if you see. So that is also you can see through this figure. And the neurotransmitter that is released from this postganglionic neuron of the parasympathetic division, it can be either acetylcholine or nitric oxide, NO. Okay, that will go and stimulate the target organ, and target organ it can contract. So this is for the parasympathetic division. If we talk about the sympathetic division, so that time the preganglionic neuron, soma is in the spinal cord, it is releasing acetylcholine. It will go and stimulate the next postganglionic neuron. So here the soma is a sympathetic ganglion located next to the spinal cord. So this is also present very next to the spinal cord. And for this postganglionic neuron, the neurotransmitter that is released is norepinephrine. So we have epinephrine and norepinephrine. Okay. So the neurotransmitters releases norepinephrine and it will go and stimulate the target organ. So this is for your sympathetic division. Okay. So this figure is important. Try to remember what type of neurotransmitters are released. So now in the autonomic nervous system, these are the fibers that run from the ganglion to the effector organ, the postganglionic. Okay. Cholinergic. Okay. Then there are two more terms. Okay. Cholinergic is these cholinergic neurons will release acetylcholine. Remember. Okay. While adrenergic will release adrenaline. So wherever the neurons are releasing acetylcholine, we'll say they are cholinergic. Okay. If adrenaline, epinephrine, that is, we'll call them as adrenergic. Is it clear till here? This diagram is clear. So you need to little bit memorize the name of the neurotransmitters, then it becomes easier. Okay, then okay, Ashwarya. So again, I would like to share about the Raftar batch. Already I've given the details. So just share it among your friends and whoever needs to prepare for CSIR NET exam, so that they can just join in time, so that they can prepare well in advance before the exam. Thank you everyone. Thank you for being such good listeners. It was good interacting with you all. So stay tuned for tomorrow's session. I'm sure it will be more exciting and we'll learn more through these interactive sessions. Thank you everyone. Have a great evening. Bye students.